Okay, so I'm going to try and demonstrate this as best I can. This is the first attempt I've ever made at it, so I'm going to do the best, as I said, best I can. So you're going to have this Ziploc bag. You have your strawberry. If you have any green stuff on the strawberry, you're going to want to take that off. We don't want that DNA material in there, so take all that out. You can place the strawberry into the plastic bag. Make sure the bag gets closed. Zip it so they are closed. And you are gently going to squeeze the strawberry into a puree form, basically as smooth as possible with as few lumps as possible. So I'm going to go ahead and do this for a little bit. I will be back in a few minutes when I have it down to what I think is a good example. Okay, so what I have here, if you look at it, it's pretty much a puree throughout. It's smooth throughout. I don't have any really big lumps in it. So we're going to go ahead and set that side off to the side, or if you're doing this in the lab with your group, you're welcome to have other people set this up while you get this part ready. So here's my puree form. I'm going to set this off to the side for right now. Now you have two pieces of material at your table. You have the AB tray. You can tell by the AB on it. And you have the funnel tray. If you'll notice that the funnel tray fits pretty much right on top, there's a little kind of a rest for that tray on top of it. Okay. Uh, then you have a square of double layer cheesecloth. You're going to place that into the funnel just on top of A. So we're going to use that at some point to capture the materials. Okay, at that point, one of your group members, you come up and you're going to grab 5 milliliters of the lysis solution. The lysis solution are the yellow bottles. You can use a small dosage cup. If you look at the dosage cup, on the left-hand side is cubic centimeters, on the right-hand side is milliliters. We're just going to go up to 5 milliliters. That's second line. So I'm going to gently pour that in until I get to about 5 milliliters. And then I'm going to pour that into the same bag with the strawberry. Well, the strawberry puree as it is now. Okay. I'm going to sit there and gently mix it, not crush it, just kind of mix it for about a minute. The lysis solution is helping to break open the cell membranes and nuclear membranes right now. It allows the DNA to kind of get into the solution itself. So about 15 seconds in, and all I'm doing is just gently kind of putting it all in the corner and working that lysis solution all around the strawberries. And again, breaking down the nuclear membranes and the cell membranes to cause it to, for the genetic material, the DNA, to kind of escape those areas so we can actually extract it. So about 20 more seconds. We want to make sure that when we're doing this, I have some bubbles on the top. I want to try and prevent that as best as possible. So I'm just kind of going gently. Just want to make sure it's all kind of mixing around, kneading around. But I don't want a lot of bubbles in the mix. Okay. And that's just about our 1 minute. So I'm going to pause it for a second, and I'm going to come back and explain our next step. So what you're going to do, now this is kind of open, so I'm going to go ahead and fold that again. Just kind of make it a little more difficult to get through. Very carefully, I'm going to pour what I can into tray A. So B is empty, tray A, I'm going to go ahead and leave it aside. I don't want any of the solids into the tray. So I'm going to very gently pour it in. And I want it in the cheesecloth. So the cheesecloth catches most of the stuff, or it gets all over the place. We'll just kind of stop it. We'll just call it there. Now I'm going to let that go ahead and drain. We have more, so if I need to do more, I can. Yeah, I'll make a mess in the process. I can do that. Yeah, I'm going to let that sit for a minute or two. I'm just letting it kind of drip in there. I want to catch as much as I can. The tray doesn't really hold a lot. The rest of the bag, you can go ahead and recycle. You don't need that. in a second. So once you have that done, easiest way of doing this without making a mess, without having to kind of worry about picking it up and dropping the solids in there, is the tray just kind of comes off. So you want to take the tray off of the bottom tray, just put it off to the side. Maybe someone in your group can rinse that out. What you have left is just the strawberry DNA and the lysate, which is what the DNA is. So we are going to remove the funnel tray, which we did. We are going to put this solid, we have this clear tube. We're going to place that into the other side B. You have a small test tube. You're going to place that in on an angle because you're going to want to drip the material in. We're going to measure out 5 milliliters of DNA solution, a DNA precipitation solution, DNA precipitation solution, and we're going to put that in the test tube. So this is the DNA precipitation material is the orange bottle. That will help kind of pull it out even more and allow it to be observed. Again, I just want 5 milliliters of this, so I'm going to measure up to that second line once again. I apologize, you can't see it. Kind of moved it away. All right, so I have 5 milliliters. The tube does, or the measuring cup does bend. So what you're going to do is just very carefully, you're going to pour that DNA precipitation solution into the test tube and have someone rinse that out for a future class to be using later on. Cap that up. Now, you have an eyedropper. What you're going to do is using this stuff, the lysate. You're going to draw it up, and you're going to put 15 drops. Now, you don't want to just drop them in. You want to kind of slide them down the side. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15. Okay, so we have 15 drops of the lysate into the test tube. You want to slowly slide down. Now, we're going to let this sit for 4 minutes. Now, I'm not going to let you sit for 4 minutes. We're going to come back. So I'm going to pause it. In the process, I'm going to clean up. So this will be a good time for you to clean up and start looking at getting ready for the last part of the experiment. So we're going to let this sit for 4 minutes. All right, we're about 2 minutes in, but I wanted to kind of show you as best I could. I shouldn't really disturb this, but I want to kind of show this close up. So if you look at the actual, if you look at the actual material, you can see that it's the DNA material is starting to rise up. I don't know how well focused it is. I apologize. But you can see the DNA material is starting to rise up in the test tube itself. And again, while you're doing this, this would be a good time to kind of clean up and get your sorry about that abrupt cutoff. We're just about 4 minutes. So at this point, the DNA should have risen up out of the lysate solution and into the precipitation solution. So that 10 times fast. I'm going to take my wooden stirrer. I'm going to gently place it in, and I'm going to kind of try or drop it. But I want to catch all those little strands. I got them. It was not pretty. Now, I'm going to put these on a slide because I actually want to kind of see what's going on with the DNA. But if you can kind of see the white material at the end of it, that is the DNA. So let me try it with another one. I'm probably not going to get as much this time, but it's the price of being clumsy, I guess. You just want to kind of twist it around a little bit. And you can see there's a strand. I know it's hard to see, sorry. But that strand is the genetic material that we're looking for. So I'm going to put that on a slide. Now, you're not going to be able to see the actual specific DNA, but I'm always one that wants to try and see what stuff looks like close up. So I'm going to go ahead and look at the microscope and see how this looks. Your cleanup instructions are basically to go ahead and empty all the chemicals, rinse out all the containers, put them back on the towels on the side of the station, and then we'll go back, we'll do the data, and we'll answer the questions. I hope it was helpful. I never had done this before, so I'm glad that I got a chance to kind of work through it and see that it does work. So I wish you luck in your DNA extraction.